map biological pathways by which social environments influence gene expression by viral, cancer, and immune cell genomes. He has been the pioneer of the field of social genomics, and I think it all started back in 2007 when he was looking at older adults and social connections, and he was able to map out that social connections were actually related to immune cell gene expression among healthy older adults. Fast forward to 2019, and this field has really taken off. The field of social genomics now really represents for us a viable mechanism by which social environment, behavior, and emotions link biology and to disease risk and activity. Much of the work that Dr. Cole has conducted is in cancer models, so very relevant to the work that we do here. He's an exceptional researcher and a colleague that has been recognized by multiple awards. He has an enviable record of high-impact publications and is in multiple grants, and NIH, and the NIH and particularly the NCI highly values the work that he does in social genomics. Interpersonally, despite his exceptional achievements, Steve is a very humble and approachable scientist, and I've been very fortunate to know him for many years and collaborate with him. So please join me in welcoming Dr. Cole.
Thank you, and thanks for the opportunity to come talk with you guys. I am going to tell you today a little bit about a field of research that has really sort of sprung up even more recently than the social genomics that Frank was talking about, looking at how nerves interact with cancer cells and the implications that has for how psychological and social processes might influence the initiation and progression of cancer. So this notion that psychological or social processes are somehow relevant to what's happening in the context of cancer is not, as you guys know, a new one. It stretches back at least to the 2nd century AD and the Greek physician Galen, who, when he was still kind of describing cancer as a phenomena, came to believe that it was particularly likely to afflict people who had certain characteristics in their personality or certain conditions in their life. So for instance, Galen thought that people were particularly likely to get cancer if they were melancholic, if they felt sort of sad or anxious or other forms of negative affect. And that idea that the social world or our psychological processes somehow influences cancer development has kind of woven in and out of fashion over the centuries since then. As you know, that's actually greeted with quite a lot of skepticism, I think, by mainstream cancer biologists these days, in part because they can't really figure out how emotional processes or stress or other kinds of, sort of psychosocial factors would interact with the physical, let's say, chromosomal damage that we think sort of drives the initiation of cancer, or the microenvironmental biological processes that, sort of contribute to the progression and metastasis of cancer once it's been initiated. That's actually what's changed in the last maybe 5 to 10 years worth of research, is we've gotten a much better set of answers to mainstream cancer biologists about how that might actually happen. So that's what I'm going to talk to you guys today about. We got interested in this area of research, not just myself, but the other folks working in the field, mostly motivated by data from epidemiology, showing that regardless of what cancer biologists think, given their paradigm about how cancer works, somehow something must be happening to connect the social world to the biological processes driving cancer progression, because it is hard to find any form of cancer that doesn't show, for example, a 5 to a 15% difference in 5-year survival rates as a function of social resources or social vulnerability. So, for example, across just about every type of cancer out there, people of higher socioeconomic status live longer than people of lower socioeconomic status. It turns out even if you control for differences in treatment, there are still those differentials present. Similarly, people who are married or have good basic sort of social support and relationship resources to help them through the cancer experience, they live longer as well. Again, not a small amount, 5 to 15% more people living 5 years or longer, and sort of a similar, with several years worth of differential survival time as a function of these social resources. So there's an effect there of some sort, and it's big enough to be worth trying to figure out so that we can try and essentially distribute that beneficial effect to everybody. And these effects, it turns out, aren't just restricted to the most, if you will, sort of puny or sort of easy to manage cancers, the ones that have sort of good 5-year survival rates. So I've plotted the relative aggressiveness of cancers on the horizontal axis there. On the vertical axis, I'm plotting average survival times. This is all out of several million cases of data from the SEER registry. And the average survival times in general for people with low levels of social resources are in the black dots, and the arrows that extend up from those are the extent to which 5-year survival increases above that sort of basal condition for people who have favorable primary relationship status. So they're married or in a secure relationship. And you can see that there's certainly, sort of increments to survival time on the sort of upper right-hand corner of that diagonal. In other words, for cancers that are pretty survivable, large numbers of people survive 5 years or more, you can see, a fair number of arrows going up a noticeable distance, some in the intermediate range going up quite a lot, maybe as much as sort of 10%. But what's important to note is even for the highly aggressive cancers in the lower left-hand corner, these are things with very poor 5-year survival rates in the range of maybe 10 to 30% or something like that. Even there, you see significant increments to survival. So it's not the case that these effects are occurring only for cancers that are sort of, if you will, on the fence and could be sort of easily pushed one way or another by therapy or by sort of lucky neuroendocrine conditions. Even really aggressive cancers show these kinds of social and psychological gradients. So we've gotten quite interested as a field in understanding where that comes from. And one way of attacking that is to go into these patients and actually look at their tumors and say, what is this tumor doing differently that allows it to progress more rapidly in people who are socially disadvantaged? So in those kinds of studies, for example, transcriptome profiling studies identify literally hundreds of genes that show differential activity as a function of patients' life resources. So for instance, in this particular study, we're looking at gene expression profiles from women with ovarian cancer. These are the ovarian tumors themselves. It's a very exciting presentation venue because the lights keep going up and down. So keep me awake, I guess, hopefully. So the heat plot there basically shows that for people with high levels of depressive symptoms and low social support, again, there are hundreds of genes that are showing differential activity levels in these primary ovarian tumors. So that's the kind of stuff that helps us understand a little bit about how those social factors might translate into different survival times. In other words, if the biology of the tumor is different, even when, in this case, we've held constant things like grade of tumor, stage of tumor, its kind of pathological conditions, everything that we would normally work up in the pathology of an ovarian cancer turns out to, these effects emerge above and beyond those variations. So when we do bioinformatics, we sift through these gene expression profiles and try and figure out, essentially sort of thematically, what's going on. What are the processes that are functioning, let's say, more aggressively in the ovarian cancer from a woman with low social support and high depressive symptoms? So for example, one of the things that comes up over and over is we tend to see more activation of this sort of epithelial mesenchymal transition dynamic. There's a greater sort of mesenchymal phenotype in these tumors from women with low social resources and a sort of comparative reduction in epithelial gene expression profiles. So that's one example of a biological theme that we kind of pick out of these, what are still fundamentally correlational studies where we're looking at patient tumors, asking descriptively how they're different as a function of these social conditions. But we'd really like to understand how those differences come about. What's the kind of causal architecture that leads psychosocial processes to potentially change the activity of these tumors? So for that kind of research, we go into preclinical models, particularly mouse models. Those have really transformed our ability to answer these questions about what are the processes through which psychosocial factors could potentially influence cancer biology. When we did cancer biology in test tubes, just the cancer cell, it was, we could see these effects sometimes in certain types of cancer, but it was kind of ambiguous. We weren't seeing the effects of the size and the consistency that would explain the epidemiologic data that I showed you earlier. So it's when we put tumor cells into living, breathing organisms that we really start to see these effects emerge more prominently, more consistently, more reliably, and it becomes easier in these preclinical models to dissect the mechanisms involved. So we take, for example, standard paradigms where you have, let's say, a mouse, and you inject, in this particular case, we're injecting breast cancer cells from the mouse back into the mouse, into its breast tissue. And we've tagged those breast cancer cells with a firefly gene so that we can take the mouse and put it in a dark box and use highly sensitive cameras to trace where in the body the tumor cells are. And so in this particular scenario, you can see that kind of purple focus, I think, in the kind of lower right quadrant of the mouse with a kind of maybe a blue centroid to it, the area where we initially injected these cells. This is a day or so after we injected the cells. You can see there's a tumor that's essentially kind of taken in that environment. And we can track the growth and distribution of that tumor over time by using that highly sensitive, sort of firefly luciferase imaging. So in this particular case, you can see the primary tumor get larger and larger over time, and eventually, somewhere around maybe day 20, 24 or so, you can start to see the appearance of another focus under the mouse's armpit. That's probably its lymph node. There's a bunch of tumor cells that have escaped from the primary tumor and transited through the blood or lymphatics and colonized some kind of a distant tissue site and are now growing a second tumor there. Basically, this is the process of metastasis. Of course, this is what we really worry about in clinical oncology. Usually, we're pretty good at containing primary tumors, but it is not an easy job to contain, sort of fulminant metastasis. That's usually what drives morbidity and mortality in cancer. So with this kind of a basic mouse model of how cancer is working in a whole organism, we can do some fairly simple experiments where we ask what happens if that organism leads a chronically stressful life. So in this particular case, we can take some of these mice that we've given these breast cancers to, and we can give them a moderate degree of stress for a couple hours a day, each day, for maybe the first week or two of the study, essentially after we've injected the tumor cells. The way we do that is we take mice which love to run around and we put them in a small box where they can't run around. Mice want to run around, so if they can't run around, they start scrapping at the corners of the box and trying to figure out how to get out and get out to someplace they can run around. So they're not like profoundly, overwhelmingly stressed. They're kind of agitated and anxious, much the same way I am when I'm trying to drive to work or get grants. So it's not really a sort of like a model of wartime or some profound famine or something like that. It's mostly a model of what behavioral scientists call hassles. So we hassled the mouse for a couple weeks in the beginning of this thing, and what we typically see is something like this. In a mouse that's not hassled, you see, so there's a modest degree of metastasis. On the average, in a mouse that is hassled, you see something like that. You see more distant metastases, and usually larger distant metastases. Interestingly, if you look in the lower left quadrant of the mouse at the primary tumor, you'll realize the primary tumor is not noticeably larger in the stressed mouse, and that actually is a fairly reliable result. When we run these kinds of studies, the primary tumor does not generally grow faster. Sometimes that happens, but usually not. What happens is the primary tumor becomes a much more efficient metastasis seeding machine. It throws off cells, and those cells have a better chance of surviving their transit through the blood and lymphatics and successfully colonizing some distant tissue site. So this is the kind of sort of the respectable version of that, where we've done it in a bunch of animals and averaged. That's the growth of the primary tumor. You can see the stressed animals really don't show any sort of noticeably larger primary tumor. It's not growing at a dramatically greater rate on the average. But if you map the mass of distant metastasis, you can see there's this exponential acceleration of metastasis in the animals that are hassled for a couple of weeks. So the good news about these mouse models is that, of course, we can very industriously and elegantly dissect the biological mechanisms involved. And I'm not going to take you guys through primary data because thankfully this literature is now so large that it would take me days to go through all of the studies that have shown this kind of thing. So what I'm going to do instead is show you the 3 general themes that have come up when we do biological dissection of these kinds of stress paradigms in the context of these mouse preclinical models of cancer. So the major ways that this experience of threat or stress or hassle makes its way to the tumor, so you've got a brain up there experiencing stress, threat, or hassle. You've got the tumor illustrated with the blue cancer cells and some kind of interdigitating microenvironmental cells like blood vessels and macrophages and stuff like that, sort of in the sort of center left of the diagram. What this is a sort of a schematic of the flows of information through the nervous system that eventually reach the tumor. One of these flows involves the innervation of the tumor itself, and I'll spend a fair amount of time talking about that today. But basically, nerve fibers, we have discovered, actually reach tumors. They generally don't do that by kind of swimming by themselves through the body and navigating and finding a tumor and sort of arborizing and synapsing. What they're doing in general is piggybacking on blood vessels. So when tumors get larger, they need more resources. That generally creates a kind of hypoxic drive for the production of more blood vessels. And these blood vessels arrive with sympathetic nerve fibers in particular piggybacking on them. So these are fibers from the sympathetic division of the autonomic nervous system, usually carrying norepinephrine. These nerve fibers in the environment of a tumor often arborize because tumors often secrete growth factors and other kinds of resources that are effectively neurotrophic. They help nerve cells home towards that particular tissue environment, and they support the arborization of these nerve fibers. So one of the ways information gets from the brain down to the tumor is via sympathetic nerve fibers that piggyback on blood vessels as they're recruited into the tumor microenvironment. A second way involves the nervous system regulating other organs in the body that produce resources that then help fuel tumor growth and metastasis. Some of the areas this is particularly prominent are innervation from the autonomic nervous system that goes into fat tissue. So you've got adipocytes making a wide variety of factors that end up fueling tumor growth, as well as organs like the liver. So you've got the production of things like glucose, adipokines, things like that end up being key resources to help cancer grow, survive, thrive, and metastasize. So when the nervous system modulates these systemic resources, that becomes another pathway through which experiences of adverse life circumstances can end up affecting the prognosis of the tumor at this kind of key biological level. And then a third mechanism involves the role of the nervous system in regulating the development and ongoing sort of homeostatic recalibration of the immune system. So you've got nerve fibers, it turns out, entering all of our primary and secondary lymphoid organs. So in the bone marrow, for example, one of the things we know is these sympathetic nerve fibers are actually key elements of the hematopoietic stem cell niche. And the nerve fibers end up releasing norepinephrine. That signals hematopoietic stem cells to lighten up on the production of lymphoid lineage cells like T cells and B cells, which, all things equal, we'd actually like to have more of those because they tend to be more often associated with effective anti-tumor immune responses. You get more myeloid cells coming out of this bone marrow that's in a body under threat. These myeloid cells, particularly the monocytes, get recruited into the tumor microenvironment. As the tumor's getting bigger and it's squashing its neighbors and they're spilling their guts, that is a big recruitment and activation signal for these monocytes. So you get these cells being born in the bone marrow from this greater stimulation from the sympathetic nervous system, and then essentially more kind of push of these myeloid lineage cells into the tumor microenvironment, and that ends up promoting metastasis. As you guys know, these myeloid cells in particular have multiple pathways through which they can facilitate tumor progression and metastasis. This includes changing the cytokine profile in a way that makes the cellular immune response less effective. They also will execute these local wound healing responses that effectively end up driving that epithelial mesenchymal phenotype that I described earlier. So that's actually, as far as we can tell, the major pathway through which that EMT shift happens, is you get more of these, if you will, sort of stress-conditioned monocytes coming into the tumor microenvironment and interacting with the growing tumor. So, it used to be thought there wasn't much innervation of tumors. We sort of bumped into this when we were, sort of looking in tumor biology models, and actually sort of relatively recently, just within the last 5 or 10 years, people started doing classical neuroscience studies where you would use, for instance, fluorescent tracers for catecholamines and finding, wow, there's a ton of catecholamines in these tumor tissues, and they tend to be around the lumen of a blood vessel. So that's, we ended up being very confident that much of the innervation in these tumors is riding in on blood vessels. And we, since then, were able to very quickly map out what are the implications of having more of this nerve fiber-delivered norepinephrine in particular for the growth and differentiation of these cancer cells themselves. So schematically, what happens is you've got these sort of tumor cells, which all kind of cartoon with that lipid bilayer. The tumor is essentially, the tumor cell is kind of sort of south of that on the diagram. North of that is, circulation, some blood vessel that is going to deliver catecholamine neurotransmitters, either norepinephrine from those piggybacking nerve fibers, or sometimes at levels of higher stress, epinephrine circulating through the blood as a hormone after being released from the adrenal gland. In either case, those signals end up interacting with beta-adrenergic receptors. Beta-adrenergic receptors are present on a number of cell types. I'll show you one schematic here about what they do when they're present on the tumor cells themselves. Those beta-adrenergic receptors, again, pick up the signals either from hormonal epinephrine or nerve fiber-released norepinephrine, and that kicks off a classic second messenger signaling cascade that eventuates in the activation of a wide variety of transcription factors and other intracellular signaling pathways that end up collectively changing the expression of those hundreds of genes that I was describing earlier in that ovarian cancer example. In general, when you get these cyclic AMP, protein kinase A, and EPAC signaling pathways activated, the classic handmaidens of the beta-adrenergic receptor, what you see as the transcriptional response, the wiring diagram of transcription factor activation, ends up stimulating the production of pro-inflammatory genes and angiogenic genes, things like IL-6, IL-8, vascular endothelial growth factor, matrix metalloproteinases, prostaglandin synthases, particularly PTGS2, and stimulating the activity of these EMT master regulator transcription factors like SNAIL2 or the slug transcription factor. Simultaneously, these pathways suppress the activity of some of the key mediators for cellular immune responses. These we see sometimes in tumor cells, more so in immune cells, but particularly suppression of type 1 interferons, type 2 interferons, interleukin 12, all of the things that we need to power the production of a good, strong T cell response. Those tend to be suppressed by this cyclic AMP protein kinase A signaling pathway that gets stimulated by these noradrenergic interactions with the beta receptor. There's a set of effects that are mediated both by those transcription factors and the suppression of the sort of cellular immune response genes and stimulation of these kind of innate immune genes. There are other effects besides these transcriptional mediations that also play a role. For example, you get changes in cytoskeletal activity that result in cell motility, particularly through focal adhesion kinase. You get changes in the phosphorylation of cell survival mediators and cell death mediators that end up making the cells essentially more, if you will, zombie-like and resilient to things like chemotherapy and other factors that would normally help cull the cancer cell population. So in these models, preclinical models of sort of cancer in mice, either mouse cancers in mice or human cancer cells in mice, what we see in general is kind of a syndrome across a wide variety of different studies at different sites in the body, sort of orthotopic models of lung cancer or ovarian cancer or breast cancer. Basically, we start to see across lots and lots of different studies a relatively consistent set of themes in animals that are stressed. You see more norepinephrine in the tumor itself. You can actually literally take the tumor out, run a catecholamine assay, and there's norepinephrine in there, and there's more of it in a stressed organism. Most of that is associated with greater density of innervation, so you can come up with the same result if you just map the density of nerve fibers in there. Generally correlating with greater innervation is a greater number of macrophages, particularly activated macrophages, and those macrophages tend to have higher polarization towards this kind of reparative wound healing phenotype, the sort of M2 or M2-like, and generally undermining the sort of differentiation towards this more cellular immune supporting macrophage phenotype, more M1-like. As a consequence of that, we generally see more EMT, epithelial mesenchymal transition, that promotes metastasis in a wide variety of ways. We see more vascularization, vascular endothelial growth factors recruiting more of these blood vessels. We also see more lymphovascularization, as my former postdoc Erica Sloan discovered. You get the growth factors required for lymphatic vessels also activated by this kind of thing. And as a consequence of all of that, you get markedly more metastasis out of these primary tumors. What we don't see in general are big changes, for example, in the fundamental cell growth circuitry that controls the rate of primary tumor cell growth. As I described earlier, there's clearly some aspects of primary tumor biology that are relatively untouched by these dynamics. It's mostly these pro-metastatic dynamics and these interactions with the tumor microenvironment that are sort of the most prominent and consistent targets of these kinds of effects. If you're interested in this kind of thing, there was actually a nice little blurb written up about this field in Science magazine, probably about a week and a half ago, by Kelly Servick. And what she was doing is basically trying to sort of give a snapshot of where this field is at, both the folks who have been looking at the interaction between nerves and cancer motivated by stress and psychosocial influences, and the people who are separately looking at the role of cancer in chasing down nerves, particularly for paraneural invasion, which is a big morbidity driver in a wide variety of cancers. So what she was summarizing here is this kind of perspective that I've sketched out, where it's clear that tumor cells recruit nerve fibers somewhat through angiogenesis, somewhat through the expression of molecules that are effectively neurotrophic factors. These nerve fibers, as a consequence, help support the growth and survival of tumor cells. They modulate the anti-tumor immune response, particularly through macrophages and T cells, and they recruit new blood vessels. So if you're interested, you can take a look at that, kind of a nice mention for this field. When we look inside the primary tumor microenvironment, we ask, where are these beta-adrenergic receptors? Who in this tissue is actually listening hardest to that norepinephrine released from the sympathetic nerve fibers? It tends to actually be found most prominently on cells that label for macrophage markers. So some tumors have a decent number of beta-adrenergic receptors on them. Lots of tumors don't. So that turns out, though, not to be a very reliable sort of indication of whether the tumor is going to be subject to these kinds of effects, because many of the effects seem to be mediated by immune cells that are coming into the tumor itself and essentially changing their behavior as a function of this kind of neural signaling. Interestingly, when we did these pathological studies on tumors from mice that were in these stressed conditions, we found that not only were there sort of beta-adrenergic receptors on the macrophages, but there was more macrophages in general in the tumors from the stressed mice. So you can tell from the kind of the density of the red in the middle panel there in the stressed mouse, usually maybe 2 to 3, sometimes 4 times as much sort of macrophage cell count. So one of the things stress does is kind of enriches this myeloid cell interaction with the tumor. And that is due in no small part to the effects that I described earlier, where the nerve fibers in the bone marrow, completely different location from the tumor, are interacting with the hematopoietic stem cell system. This is flow cytometry on bone marrow cells, and you can see, in particular in the far right side there, in an animal that's subject to repeated social stress, you see more granulocytes and more monocytes coming off of the, sort of the hematopoietic stem cell production process here. So you get this shunting that actually we understand pretty well now how that's mediated by the particular growth factors that control cell differentiation during hematopoiesis. So that ends up skewing towards the production of a certain lineage of white blood cells in the bone marrow. Those cells go out into the circulation. One of the things that they've discovered recently is those cells will also colonize the spleen and start up a new blood cell production factory as sort of a temporary encampment in the spleen. This is work that was done by Daniel McKim and John Sheridan. Really interesting that chronic stress seems to create this sort of emergency myelopoiesis factory in extramedullary tissue. And as a consequence of that, you get a higher density of these monocytes in blood. And that's what ends up supplying more monocytes into the tumor microenvironment and where you get that higher density of macrophages. So we know these monocytes play a critical role in these stress effects because if we give these mice drugs that block that sort of stress-induced myelopoietic factory, you largely knock out the sort of stress-induced metastasis of these tumors. So we can do that by blocking things like macrophage colony stimulating factor or other factors that are involved in either the hematopoietic production of these monocytes or the recruitment of these monocytes into the tumor microenvironment. So pretty substantial diminution of these stress effects. So what I've shown you is mostly this kind of diagram here where the environment of a person's life ends up changing the way their brain functions. Essentially, they feel more threatened and insecure and uncertain. And one of the consequences of those changes in brain function is more activity in these descending sympathetic nerve fibers that end up interacting with a wide variety of tissues, including in the bone marrow, interacting with the hematopoietic stem cell niche and pushing this differentiation of stem cells towards a more sort of monocyte-rich result. And as a result, those monocytes go out into circulation. They've interacted, in this case, with a solid tumor, but it turns out those monocytes also probably contribute to a wide variety of other stress-related pathological processes. They get in the brain by themselves. They don't cause a lot of trouble, but if you get a brain insult with these highly abundant and highly primed, immature, pro-inflammatory monocytes, they tend to produce hyperinflammatory responses in the brain. So you get more neuroinflammation and more neurodegenerative disease out of these stressed animal models. When we look in the vasculature, these monocytes play a key role in attempting to repair damaged blood vessel linings and inadvertently creating atherosclerotic plaques. And so you tend to exacerbate the development of atherosclerosis and heart attacks as a consequence of having more of these monocytes out there. They get into the lungs, and again, by themselves don't cause too much trouble, but if you get some kind of a lung insult, they tend to over-respond, and you'll get things like airway inflammation and asthma, or sometimes pneumonia as a result of these effects. And they end up providing somewhat skewed advice to the rest of the immune system as these monocytes differentiate into macrophages, collect antigens, and as their cousins dendritic cells end up interacting with the tumor microenvironment and carrying antigens back to, let's say, the lymph node to initiate an immune response, they end up doing that with a skewed cytokine profile. So this signaling ends up essentially sort of pushing them towards a more sort of Th2 kind of cytokine profile that is good at supporting, sort of extracellular attacks on extracellular pathogens, but skews away from a sort of more Th1 profile that we'd really like in the context of cancer and viral infections, because that's what it takes to generate a good, strong cytotoxic T cell response. As a consequence of that, in general, we've seen in the immunotherapy literature that, as we know, immunotherapies work maybe a third of the time under the best scenarios. You need a good existing cytotoxic T cell response to disinhibit with these checkpoint inhibitor drugs. If there's no cytotoxic T cell response there, you can disinhibit all you want, and nothing is going to be there to, sort of come to your rescue. So there's a lot of interest in what can we do to maximize the vigor of the cytotoxic T cell response, sort of in tandem with these checkpoint inhibitor drugs. And one of the things that people were able to figure out from these preclinical models in mice is that actually one of the things that leads to the absence of a good, strong cellular immune response is a lot of sympathetic nerve fiber activity that ends up producing that kind of Th2 cytokine skew. You're sort of deficient in Th1 cytokines, and as a result, you don't generate very strong cellular immune responses in the first place. So they are able to actually block they're able to essentially resuscitate the effectiveness of checkpoint inhibitors by giving animals that have been stressed beta blockers, so that you disinhibit the cytokine support for the production of the cytotoxic T cell response, and in tandem then maximize the efficiency of that response with the checkpoint inhibitor itself. These effects are driven in large part by changes in type 2 interferon activity, and as a result changes in cytotoxic T cell killing production and killing capacity, so that you get more T cells produced and they have a stronger effector response literally at the binding stage where they interact with a target cell. So that is sort of a synopsis of what we've been able to find from this literature about what in sort of human physiology or mouse physiology in general interacts with the nervous system to facilitate cancer progression. There's one other important player in this system. There's about maybe 20 to 30% of cancers, there is some kind of an infectious agent that contributes to the initiation of the cancer. So these are like virally associated cancers, rarer. Sometimes you'll get bacterially associated cancers. Those two, it turns out, are somewhat sensitive to these dynamics, in part because many viruses have evolved to acquire a sensitivity to this beta adrenergic signaling pathway. And the reason will actually make sense when you think back to what I talked about earlier. One of the things that the nervous system does is it suppresses particularly type 1, also type 2 interferon responses in the cellular immune system. Well, if you're a virus and your chief selective pressure comes from the cellular immune system, that would be a great time to wake up and start to replicate, especially if you're like a herpes virus and you spend most of your time hunkered down in some kind of a, sort of latency state, and you need to kind of counter program against the body's immune response and wake up and replicate as fast as you can, disseminate, get back down into a new set of host cells and kind of lock back down your genome so that you can hide out from the cellular immune response. That's the general, sort of strategy that these big, smart herpes viruses pursue. And so one of the things they do is they've generally acquired a genomic sensitivity to the cellular signaling pathways that get activated by this beta adrenergic signaling, because that's a reasonably good forecaster of an impaired impaired innate immune response against viruses, and to some extent, lagging that in impaired cellular immune response. So, like I said, these cells, this is a particular virus involved in Kaposi sarcoma. We bumped into this before we were even working on cancer, when we were focused mostly on HIV infection and why it seemed to be differentially active as a function of nervous system activity. So this is a classic opportunistic pathogen in the context of HIV infection, Kaposi sarcoma-associated herpes virus. As I described earlier, big, smart virus, lots of pulleys and gears and sensors and stuff like that, that allow it to have these two lifestyles. One of latency, where there's very little gene expression going on to try and, sort of avoid immune surveillance. And then periodically, these viruses wake up and replicate like crazy. They go into what's called active or lytic replication. And one of the things that's really efficient at kicking a latent herpes virus into lytic replication is exposure of the host cell to norepinephrine or some of these other adrenergic stimuli. So this is one additional example of how a pathogen that itself contributes to the production of a cancer, in this case Kaposi sarcoma, would end up being sort of an intermediate between the nervous system and what's going on in the sort of, if you will, pathobiology that's driving the production of the cancer. So That is a kind of a quick tour through what we've learned about these systems. One of the things that will be apparent is that there's actually a number of different pathways from the brain down to tumor biology. In fact, it's pretty tedious at this point. Almost every month, someone publishes another paper showing another pathway through which this happens, either sort of organismically at the level of tissue, or another pathway at the level of the intracellular signaling pathways that mediate these effects. So it's unlikely that we're going to go out there and block these effects one at a time with different antagonists for each of these pathways. There's, literally, at least at this point, 20 or 30 different pathways in general and hundreds of genes that are behaving themselves differently. So this isn't a situation that probably lends itself to deployment of 200 antagonists for these specific individual molecular impacts. But one of the things that's happening here is, it's very complicated at this stage, but upstream, a lot of these effects, as I described earlier, are mediated by beta-adrenergic receptors. The way we determine that is we give stressed animals beta blockers, and that works like a charm. In not all the time, but maybe about 75–80% of the time, you can more or less knock out these stress effects with, sort of the beta-adrenergic antagonist that your grandparents took when they had a heart attack. Just propranolol will do just fine. You can do the same thing with genetic knockouts and all kinds of other fancier technology. But the upshot of that is these generic, cheap, relatively safe beta, sort of non-selective beta antagonists work pretty well in animal models. And so that raises the possibility that we might be able to cut these effects off at the level of the nervous system supply to these pathways. In other words, if we can block the interaction between norepinephrine and these beta-adrenergic receptors that are carrying this information into all these different pathways, we can get a fair amount of leverage over a number of different physiological systems from one reasonably manageable agent here. So as I mentioned, in mice, this works like a charm. If you take that stressed mouse, you give it propranolol, during the period that it's stressed, you can largely knock out that stress-induced metastasis. Notice again, you don't change the growth of the primary tumor. This stuff has very little effect on basic primary tumor growth. Where it exerts its biggest effect is in this propensity to metastasize. So with these observations, epidemiologists started asking whether people who happen to be on beta blockers at the time that they're diagnosed with cancer, whether they showed a more favorable course. And this is a kind of a mixed literature. As you guys know, epidemiology is tricky because beta blockers don't rain from the sky randomly on people. People get, sort of some kind of a disease that indicates the prescription of a beta blocker. And so it's difficult to, sort of carve out whether this is due to beta blockers themselves or whether it's due to some kind of confounding by indication for the initial prescription of the beta blocker. But on the average, in these sort of observational epidemiologic studies, they're typically seeing relative risk ratios for, sort of survival and recurrence that are on the order of maybe 0.5. So a 50% reduction in risk is, probably worth going after. That may not be a perfect estimate of it, but that was enough to kind of give people a sense that given the mass of the preclinical animal model data and this sort of at least a few promising hits from the epidemiological data, it's worth actually trying to do this right and go into humans in clinical cancer and try some kind of randomized controlled experiment to see what's going on. So there's now a number of those studies taking place over the last, at this point, 5 years or so. There's a bunch of very small kind of single-arm, let's give it a try studies that I'm not going to talk much about. Instead, I'm going to talk about the things that are more probative, which are the randomized controlled kind of proof of concept biomarker trials. So these are studies where you randomize to placebo versus beta blocker, often during some kind of highly leveraged window of opportunity. In the context of breast cancer, we do this with people who've just been diagnosed with breast cancer. They're going to have surgery in maybe a week or two, and we put them on beta blockers. We try and get them up to a decent level of beta blockade at the time of surgery, in part because that's a period of substantial psychosocial stress, but also in part because when you start cutting bodies open, they take that as an invitation to be stressed. So they start producing a bunch of prostaglandins and catecholamines, and that'd be a great time to really sort of protect the system from this high level of sort of autonomic signaling that is associated with surgery itself. In some of these studies, we also supplement with COX-2 inhibitors because we know that prostaglandins ramp up during surgery, and the prostaglandins actually end up signaling through a receptor system that activates the same intracellular second messenger system. So we want to make sure that we've blocked all of the inbound routes for that particular system. So I'll show you a couple of examples of that kind of window of opportunity trial concept in breast cancer and colorectal cancer. These are studies done by Shamgar Ben Eliyahu in Israel. What he did is, like I said, give people beta blockers and nonsteroidal anti-inflammatory drugs right when they're diagnosed, so a period of high psychological stress, and keep them on the beta blocker and the COX-2 inhibitor through surgery for about a week or two afterwards. After surgery, they start tapering down on the beta blocker so they don't have to go cold turkey and suffer the sort of the regulatory side effects that happen when you do that. So he gets them through surgery, makes sure that they're stable, and then sort of ramps them back down. So they're on beta blockers and COX-2 inhibitors for usually about 2 to 4 weeks or something like that. Not a very substantial period. But what's nice about that model is that we can take the resected tumor tissue and ask, for that week that they were on beta blockers, did anything unfold differently in the tumor tissue? Was that enough time to change the activity of some of these molecular pathways that we think might be mediating these effects? So I'll show you a couple of things. When we look in the primary tumor tissue at resection after that week on beta blockers, we tend to see reduced activity and numbers of monocytes. This is mostly through transcriptome profiling that we're doing this. This is not generally done with histology. We're mostly using bioinformatics to essentially count the number of monocyte-specific gene transcripts in the tumor environment. That looks like it's down. And if there looks like a compensatory, possibly, increase in the prevalence of B lymphocytes or B lymphocyte activation within the tumor. What it's doing there, I'm not totally sure, but that's what we saw. We also saw reductions in M2 macrophage differentiation indications, which is encouraging. We know that's one of the mechanisms by which the sympathetic nervous system promotes metastasis, is by activating the M2 differentiation program. So seeing less of that is promising. And we see reductions in this kind of mesenchymal gene expression profile that we worry about as sort of a promoter of metastasis. So, we did this in, I can't remember the number, but it's something like 20 patients in each arm. So these are not trials that are powered for clinical endpoints. They're powered specifically to look at these kind of molecular proof of concept outcomes. But at least at that level, this looks reasonably promising, especially given that they were treated for a relatively short period of time. In the context of colorectal cancer, he's done the same kind of window of opportunity trial again, saw reduced indications of monocyte numbers and activation, in this case reduced numbers of B cells, again directionally a reduction in M2 differentiation, although that was not significant in this particular study, and a reduction in mesenchymal polarization. So in the context of those studies, it looks sort of pretty promising. There's at least decent molecular proof of concept data that something that looks generally positive at the molecular level is happening, enough to hopefully license the next round of trials with more hard clinical outcomes in much larger sample sizes. There's also a set of studies going on at Medical College of Wisconsin where they are particularly specialized in hematopoietic stem cell transplants for hematologic tumors. So hematologic tumors are, as you guys know, very different biologically from the solid epithelial tumors I've talked about before, but they, it turns out, are subject to the same kinds of stress-induced effects, in part because that nervous system interface with the bone marrow is part of what allows hematologic tumors to survive and progress. Hematologic tumors are essentially stem cells or their sort of differentiated progeny run amok. And so they need the same kind of physiological supports as sort of a healthy stem cell does. And so this norepinephrine signaling tends to aggravate them as well. Treat those animals with propranolol, and in general, you get reductions in the aggressiveness of disease. So in these studies, she's giving people beta blockers when they're having hematopoietic cell transplants in the context initially of multiple myeloma. So this is a trial that's just been completed. I think she's almost got her paper accepted, so should appear soon. But what Jennifer saw when she gave people beta blockers during stem cell transplant recovery, basically, is increases in activity of this is all circulating blood that she's looking at, but within that circulating blood pool, she gets increased expression of genes that are selective for CD34 cells containing the hematopoietic stem cell population, CD105 positive cells, which also include mesenchymal stem cells, reductions, and this is actually what we were shooting for, reductions in expression of genes that are characteristic of myeloid-biased progenitor cells. What we're really worried about is the sort of the stem cell compartment deviating by virtue of stress in a very kind of pro-myeloid way, and as a consequence of that, under sort of recovering T cells and B cells that we need. She also looked at monocyte differentiation in these circulating blood samples and again found reductions in classical monocyte gene expression, which are exactly what we would expect if we're blocking this nervous system drive for more monocyte production. So reasonably promising. So that's sort of, if you will, a kind of quick tour through what we're doing right now as sort of the first line of studies to try and deprive the cancer biology in general of sort of stimulation or support from this kind of central autonomic drive. We've been doing this, primarily physiologically. As you guys know, here at Sylvester, you guys have a very active psycho-oncology program, and that's what, on some level they're trying to do as well, is address the stress that, cancer patients and survivors experience and the effects that that might also have on residual disease. And so here the idea is to, perhaps instead of or in complement with cutting off these signals peripherally in the body using a pharmacologic antagonist, kind of move up to the experience of life in the brain and reduce the extent to which it is driving stress biology out into the periphery. And as you guys know, Mike Antoni, for example, has been very industrious in conducting these kinds of studies, testing whether cognitive behavioral stress management can change certainly psychological and neuroendocrine outcomes, and in a sort of an incidental analysis of impact, he even finds indications of better survival as a function of these kinds of stress management interventions. So that is kind of what I'll leave you with. I'll give you through kind of the quick update. I guess it's kind of a dated update now, since it's maybe a year old, but things haven't changed that much in the general summary here. When it comes to stress and cancer in clinical trials, we now have a pretty well-established preclinical evidence base that there's a recurrent role for sympathetic nervous system activation and beta-adrenergic signaling in promoting particularly solid tumor progression and metastasis, and a pretty well-defined set of tumor biology mediators. In other words, we know about the cellular mechanisms involved in these things, and so we can use those as outcomes in trials. There are, at this point, promising results from cellular and molecular biomarker trials from 3 now completed trials. There's another trial actually that just wrapped up, also reasonably successfully in Australia. These generally involve beta blockade, sometimes supplemented in 2 of the cases by COX-2 inhibitors in the context of early stage breast cancer, early stage colorectal cancer, hematopoietic stem cell transplant, and now in a second study of breast cancer in Australia. They all see fairly similar results, positive results at this kind of key molecular biomarker level. So there are some significant, limitations. I won't sort of claim that, hey, we've got this one licked. There's no data yet on survival from any of these kinds of interventions. We're not anywhere near powered for those kinds of studies. So, whether these biological effects are big enough to create meaningful clinical outcomes remains to be determined in future studies. Usually about 20 to 30% of the people that would like to be on these trials end up being excluded because there's some kind of a contraindication for beta blocker treatment in them. So beta blockers aren't perfect as a, sort of like you can give them to everybody without worrying about it. So, usually maybe 15 to 20% of people have a contraindication. Another maybe 5 to 10% try to go on the beta blockers, but have some kind of a, sort of a adverse reaction. We just can't get them up to a very high level of beta blockade before they get hypotensive or something like that. And so for that reason, there continues to be great interest in not just pursuing pharmacologic attacks on these kinds of issues, but through behavioral interventions to try and limit these effects as well. That said, we still don't have behavioral interventions that have been, implemented a priori to target clinical outcomes as their disease endpoint. So we have plausible protocols for doing that kind of stuff, but we don't have studies yet where we've said ahead of time, this is our primary outcome, this is what we're targeting, and it turns out that's actually what worked. We have lots of sort of incidental retrospective analyses saying, hey, the people that were in the stress management intervention definitely, survived longer, but this is not, what the trial was originally put together to look at. So that's what I'll leave you with, this notion of leveraging neural influences on the tumor microenvironment. We know that the tumor, is composed of lots of cell types and influences from local non-tumor cells as well as the tumor cell itself. What we understand more broadly is that all of the cells in the body are under some degree of physiological regulation by the brain and the nervous system. And so it's not implausible to think that those things will somehow seep down to the cancer cell and change its function. And that's really what we hope to exploit in the best possible way, the most efficient way, to help essentially remediate those social disparities where people living happy, privileged, comfortable lives end up with a less aggressive case of cancer. We'd like everybody to be physiologically enjoying the same good protection against cancer that those of us who are having the best lives enjoy kind of by default. So with that, let me thank you for your attention and note some of the people that have contributed to this, particularly Anil Sood and Susan Lutgendorf on the ovarian cancer research, Erica Sloan and Don Lamkin on the preclinical data I showed you, particularly for breast cancer, and Shamgar Ben-Eliyahu and his team in Israel for the clinical trials data, Jennifer Knight at Medical College of Wisconsin, and Paige Green from the National Cancer Institute, who is our institutional champion for this. So with that, let me thank you and I guess take questions if we still have time.
Apologies for sound effects and light shows. Sounds of progress. We have time for a couple of questions.
Yeah.
So I'm a psychiatrist and I treat cancer patients. So, if somebody has attempted to do research with certain reuptake inhibitors, because that's what I would use for managing anxiety, depression, and even antipsychotics for certain symptoms that cause stress or complications.
I have nothing on antipsychotics. There's very little on SSRIs in these things, but I believe there's a couple of reports of those. One of the things that's complicated about preclinical mouse models is that if you mess with models long enough, you can find just about anything. And so lots of models have these quirks. So the one caution I would say about the SSRI reports that are out there is that they were in relatively atypical mouse models. So people that are preclinical modelers know that they're like these industry standard models. And so where we saw the SSRI effects were like a little bit atypical results, things where we worry a little bit about, is this model